Assalamu alaikum dear students. Welcome to all of you in our new lecture, lecture 56. My dear students, this is class first year biology and I am your teacher Adnan Ali. Dear students, in our previous lecture, lecture 55, we discussed and explained the mechanism of opening and closing of stomata, in which two theories we explained. First one was starch-sugar conversion theory and the latter one was the potassium ion flux theory. In that two theories, we explained different mechanisms which were involved in the opening and closing of the stomata. Dear students, today I am here to deliver my next lecture, lecture 56, and our topic will be translocation of organic solutes. Organic translocation of organic solutes. First of all, we will define what does it mean by translocation. The movement of different organic solutes to different parts of the plant is called translocation. Now, according to the Ernst Munch theory, which is also called as pressure flow mechanism, according to this pressure flow mechanism or pressure flow theory, the food move or the organic solutes move from source to sink. Now, first of all, what is source? Source is the part of the plant which is preparing the food, preparing the sugar, and definitely these are the green parts of the leaf. And source, sink is the area where storage and utilization occur. So definitely it will be stem and roots. So the organic solutes will move from the source to sink. Now composition of the translocating substances. Now in the composition, about 10 to 25% is the dry matter. And 90% of this dry matter is saccharose, which is a disaccharide, which is also called as cane sugar. So table sugar also. So this is the composition. Now what is the mechanism? So for mechanism, we need to draw the diagram. This is source. These are companion cells. Zinc cells. You can see the root cells. This is U2. This is U plate. So according to this mechanism, when the glucose molecules concentration, saccharose molecules concentration increase here, it absorbs water from the surrounding tissues, absorb water from the xylem, which increase the hydrostatic pressure, and that hydrostatic pressure pushes actively by active transport that energy is provided by the cells, and these saccharose molecules are pushed towards the CO2. Now, then they move downward. Now, here unloading will occur. This unloading is also being done by active transport, in which expenditure of energy will occur, and that food will come to the sink. And the additional water which is being absorbed by the CO2 from the surrounding tissue, from the source by the surrounding tissues, they will be again sent back to the xylem, and in that way food is going from the source to the sink. And this is called as Ernst Munch theory, and this is called as pressure flow mechanism. So again, quick summary. Dear students, this is source. Source is the area of the plant which is usually preparing the food that absorb water from the xylem because our water potential is decreased. So when decreased water potential, that food or that organic solutes are pushed towards the CO2 by the help of companion cells, which will provide energy for this active transport. And this food will move through the body of the phloem or through the CO2, and when it will reach to the sink or the root cells, unloading will occur by the help of active transport, and in this way the food will move from the source to the sink. So it was all about the pressure flow mechanism. Now the next topic we have is the growth and development. Next topic is the growth and development. Dear students, first of all, we need to know what is development and growth. So development is actually a series of changes, a series of program changes in which organism is converted from simple form to complex one. And growth is increase in size. Growth is increase in size. So growth is actually the part of the development. Usually we have four phases of growth and development. The first phase is called as division. Phases of development and growth. First is division. Means cell division. Second is cell elongation. The third one is maturation. And the fourth one is differentiation. So these are the four phases of growth and development. Now dear students, first of all, the growing tissues. We will talk about the growing tissues. So the first one is meristematic cells. These are small cells having abundant cytoplasm. We having very thin cell wall. Thin cell wall they have. And just primary wall they have. Dear students, they are the dividing cells which have the ability to divide, and they divide continuously. They divide continuously, and as they have thin cell walls, so there is no problem in their division. When they divide, the cells which remains in the meristem is called initial cells. That remains in the meristem. And the later one are derivatives. Which differentiate which go into the next phases, elongation, maturation, which mature and perform different functions. Dear students, now we have certain types of the meristematic cells. First is the apical meristems. Apical meristems are the dividing cells. Are the dividing cells which bring apical growth. Now, what is apical growth? The root and shoot apexes have the apical meristematic cells. For example, this is a apical region. So here the apical meristem cells are located. They divide divide, and usually they increase the length of the branches along with the stem, and the root length is also being increased. So the basic function of the apical meristeme is to break primary growth. Primary growth means increase in length. These students, they are also called as primary meristems because primary meristems usually bring, they are called primary meristems. Why? Because they are first derived from the embryonic cells. So it was all about the apical meristem. Now the second type we have the lateral meristem. Lateral meristems. Lateral meristems are also called secondary meristems because they are derived from they are derived from the apical meristem. That's why they are called secondary, and they bring secondary growth. Mean increase in width. There are three types basically. One is called as vascular cambium, vascular meristem, which is called as vascular cambium. Which is bringing more secondary growth in the form of rings, and these rings are called annual rings because in 1 year just one ring is added. By means of annual rings we can find out the age of a woody stem also. Second one is carp cambium, which is also called as phyllogen. And they are responsible for the formation of bark, the cork tissues, stem and root. The third type is a derivatory meristem, which is usually dead cells which are left by the apical meristems when they were growing. So they are responsible for the new branches to come out. So dear students, it was all about today and about the translocation of the food and then the growth and development we discussed. Inshallah, in our coming lecture, we will discuss some more topics from this chapter. Hope that you will got good understanding. Thank you so much. Stay in home, stay blessed. Allah Hafiz.